Welcome back, everyone. So I kind of previewed this slide for you in the last lecture video that we're going to start talking about some examples of active transport that goes on in our cells. One very important one is the sodium-potassium pump, or the sodium-potassium ATPases. And this gets studied a lot. This is a type of a P-type ATPase. And in this molecule, the chemical potentials for sodium and potassium are both essential as part of that driving force for coupled transport and also for transmitting nerve impulses. The sodium glucose symporter is an example of indirect active transport. And then there's proton transport in some bacteria that is driven by light, and it's also a cool type of transport, something worth looking up, but something that we're not going to focus too much on. So in mammalian neurons, in the cells that make up your nervous system, there are about 30 potassium ions inside the cell for every potassium ion that's outside. For sodium, this ratio is, I want to say, for every sodium inside, it's like 12 outside. You can do the math to figure that out. 0.08 sodium inside for every sodium that's outside. So we are pumping both sodium and potassium against their concentration gradients, forcing sodium out when there's already plenty of sodium out, forcing potassium in when there's already plenty of potassium in. So the sodium-potassium ATPase, or the sodium-potassium pump, is going to use ATP, and right here is where ATP is going to bind, to transport both of these ions, both 3 sodiums going out and these 2 potassiums coming in. This molecule, the sodium-potassium pump, is the main protein responsible for keeping that gradient of sodium outside and potassium inside. So the sodium-potassium pump has 2 different conformational states. In one state, like this one, it's in the E1, it's open, and so on the inside, those sodium ions bind to it because this E1 conformation has a strong affinity for sodium. Then it's going to switch to the E2 conformation, and the E2 conformation is open to the outside. And in that conformation, it doesn't have as much of an affinity for sodium, so it's going to release the sodium. It has a high affinity for potassium, so it'll pick up the potassium. Then it'll switch back to this E1 conformation, release the potassium, pick up the sodium, bring it back and forth as long as it takes. So the way this works, we're starting here at step 1. You have 3 sodium ions binding to the E1 conformation, open to the inside. That is going to cause phosphorylation of ATP, of the alpha subunit by ATP. And this is going to go through this conformation shift. Now that it's phosphorylated, it shifts over to where it's open on the outside, releases the sodium, and now it's going to be attracting the potassium. So it has a high affinity for potassium. Potassium binds. That triggers that phosphate to be dephosphorylated. So now it comes back to this E1 conformation. It releases the potassium inside. Burn another ATP. Stick it on to your enzyme. 3 more sodiums. Go out. Burn your ATP. Potassium come in. Grab another ATP. 3 sodiums. Burn your ATP. Sodium out. Potassium in. Okay. So for every round, we've taken out 3 sodiums, we've brought in 2 potassium, and we've used another ATP. One type of indirect active transport is the sodium symport. And this is an additional way that we can bring glucose into our cells by using this sodium glucose symporter. This would be something that like our intestinal cells use to bring in glucose, bring in amino acids, to be able to get that energy from your foods and those nutrients from your foods, even when the concentration in the lumen of your intestines is going down. So we have that sodium-potassium pump that is keeping a very big sodium gradient. You've got lots of sodium outside of the cell, not as much inside of the cell. And that is going to provide the energy to run our sodium glucose pump. So sodium is going to come down its concentration gradient to come back in, and glucose is able to go up its concentration to gradient to come into the cell. These proteins are called sodium-dependent glucose transporters, or SGLT. For this to work, you start with 2 sodium ions outside that bind to your symporter. It's open on the inside. When those 2 sodiums bind, it also brings in a glucose. And now your glucose binds. It triggers a conformation shift, so now it's open to the inside. Sodium is released. Your glucose is released. Your sodium will probably be pumped right back out by that sodium-potassium pump, but you've got your glucose in. Your transporter moves right back to that open conformation on the exterior of the cell, and you just bring in another 2 sodiums and glucose. So you keep bringing this glucose into your cell, even when there's not a lot in the environment. Bacteriorhodopsin is something that's found in our prokaryotes. It's an integral membrane protein within the halobacteria, which are actually a type of archaea. And it has a light-absorbing pigment to it that makes it purple. So these archaea that have it are sometimes called purple synthetic bacteria, archaea. Okay. So this uses energy from photons of light to actively transport protons out of the cells, which creates an electrochemical proton gradient that allows ATP to be made. So light inputs the energy, protons pumped out, ATP synthase brings that proton back in and makes ATP. So for every transport event in the cell, these are really different types of energy transactions. When we're looking at uncharged solutes, the only variable is a concentration gradient across the membrane. But when we're looking at something charged, you have to think of both the concentration gradient and the electrical potential. If your solid doesn't have a net charge, to figure out the delta G, it's pretty straightforward. You're only looking at concentration gradients. And so you can compare the concentration outside and the concentration inside, and you can actually figure out. You can math that if you want to, but you can figure out what kind of energy change there will be for a molecule to go up or down its concentration gradient. For something that's charged, you're going to have to consider both the concentration gradient and the membrane potential. That membrane potential is almost always going to be negative. It's almost always going to have a positive overall charge on the outside of the membrane and a negative charge on the inner side of that membrane. So that usually follows that cations are going to come into the cell and anions are typically going to leave. They're typically not going to want to move in the opposite direction. All right, you finished chapter 8. We're going to come back in our next lecture videos to talk about some protein translocation. And then that is our last chapter before your next exam. I'll see you later. Bye.